Hello, my name is Eli Lyons, co-founder and CEO of Tupac.Bio. We provide software for synthetic biology research and development. In this video, I'm going to talk a little bit about biosecurity and introduce our tool BioGuard. So one of the silver linings of the COVID pandemic is that it has brought more attention to therapeutic development and research in general in biotech synthetic biology. However, I'm a bit disappointed that it hasn't brought more attention to biosecurity. So some people have put forth the notion that COVID-19 is a designed and synthetically manufactured virus. But what I think is really important is to bring awareness to the fact that we can manufacture viruses and design viruses through synthetic DNA manufacturing. And synthetic DNA just means DNA that's made in a lab. And usually this DNA is used for fundamental science research, genetic engineering experiments, but it does create the risk that synthetic pathogens can be made. So biosecurity is very important, and I'll give you 3 reasons why it's so important. Number 1, to prevent bioterrorism by individuals or groups that intentionally design or manufacture known viruses, known pathogens, for example, and want to use these synthetic pathogens for bioterrorism. One of the things I notice is that a lot of times when we talk about biosecurity, people really focus on pathogens that infect humans. But I think there's probably just as large of a threat for other types of bioterrorism. Other types would be pathogens that attack agriculture, for example. So you can imagine that there's lots of risk there. Reason number 2 that biosecurity is so important is to prevent labs from obtaining pathogenic sequences when they do not have the qualified facilities and or researchers to handle those pathogens. So typically in a research lab, you have cleanliness and safety ratings for the lab environment, for the lab room. So you wouldn't want a lab that doesn't have a very sterile environment or have the means to dispose or handle a pathogen appropriately to obtain a dangerous sequence. In addition, you wouldn't want a researcher that's not qualified. So you wouldn't want, probably your summer intern to be handling extremely dangerous pathogens when they've just learned how to use a pipette, right? That doesn't seem safe. So a third reason why biosecurity is so important is to be able to identify from DNA sequences found in a wet lab, taken from a patient sample, or from maybe a surface, viruses that or bacteria that are found on a surface, and identify them quickly and accurately and see if they are pathogenic or not, and to what degree, right? We want to accurately identify these pathogenic sequences in all those situations. Our tool, BioGuard, provides a user-friendly and secure way for users to check if a given DNA sequence is pathogenic or not by comparing it with a regularly maintained pathogenic database. And in addition to that, we provide some customization options such as custom database creation and other features around security, cybersecurity. We will also have advanced sequence screening available in the future. This will allow users to screen subsequences of a given input sequence. This video was meant to be a brief introduction to biosecurity and BioGuard. Please feel free to contact us. You can contact me at Eli, E-L-I, at Tupac.Bio. And contact us if you want to learn more about the BioGuard tool or some of our other tool offerings or what we're doing in research and development. Have a great day.